The third talk is translating mouse models of abdominal aortic aneurysm to the translational needs of vascular surgery. This will be presented by Dr. Busch, Senior Consultant Vascular Surgeon in Dresden, Germany. He's fellow of the European Board of Vascular Surgery, got MD and PhD degrees from the University of Göteborg. He performed 2 years postdoc at the Karolinska Institute in Sweden. And he's working with small and large animal models in AAA for translational research in drug repurposing and RNA therapeutics. Dr. Busch.
Yes, thank you, Dr. Diaz, for the introduction. It's very kind, and I'm very happy to present our paper. We've been asked to write a review actually on mouse models, and I'll be using that to sort of give some bullet points on our opinions, our experience here, and also share with you some work. I hope you can see my screen now. And the paper has been published in Journal of Vascular Surgery, Vascular Science in March. We're very happy about that. And just to start off, most of you are, of course, in the field of aneurysm research, so you know all that stuff, that this normal architecture within the normal aorta with the course of time changes rapidly. And of course, we have those from human samples, from open repairs, from intact and ruptured aneurysms, as my predecessor already talked about, and we know that there is fibrosis going on. There is some different hemodynamics. We've heard about possible thrombus activities. There is some proteolytic imbalance going on, possibly overexpression of the MMP9s, for example. We do see some angiogenesis going on in those aneurysms. And we know there is some sort of immune answer that might be helpful to actually condemn the process or not. So that's basically what we know from human disease, and this is of course trying to be mimicked within mice. And I guess you will all agree that we've seen a lot of good clinical studies, but this is maybe some of you have even been part of those studies. They've been reviewed a couple of times, but I guess we can all agree that there is a huge translational gap in those clinical studies, and actually taking what we know from basic science in the lab and then taking these substances to clinics. And we think that some of this might actually be due to the mouse models that are used here. And I'm just going to introduce a bit of those mouse models based on their changes to the aorta. So you can see here the normal mouse aorta, which has about 4 layers of elastic fibers, so a bit less than the human aorta. It's about 0.5 millimeter thick. Many of you may have worked with mice models before. And then I guess most of you are aware of this so-called calcium chloride model, where you expose the aorta and you put calcium chloride on the aorta. And over this course of 4 weeks, you can see some aneurysmatic dilation, some inflammation going on. We'll be coming back to this in more detail. Then, of course, there is the very famous and most frequently used angiotensin II model. You can also see a cross-section here. So after implanting this osmotic mini pump in mice and administering angiotensin II to hyperlipidemic mice, they get this dissection sort of aneurysm. You can see that in most cases the actual media is still very much intact, and we do see this large intramural thrombus, actually this large dissection going on in these animals. One model that has also been frequently used in the past is a so-called PPE model, so the perfusion with porcine pancreatic elastase. You have to surgically manipulate the aorta, which is kind of challenging, and you have to put the elastase inside the aorta. But those mice do actually get nice aneurysms, and we do see a couple of features here that are very human-like. And throughout the last 10 years, something that has come up is putting the elastase not inside the aorta, but putting elastase outside of the aorta. And you will also get this nice aneurysmatic dilation, and you can enhance those models with this cross-linking or anti-cross-linking agent. You will actually see some ruptures, you will see some intraluminal thrombus. The BAPN can also be used in the angiotensin II model, of course, and we'll also come back to that. So to make our point, and this is a very busy slide, so I will just take some time to get you through it. I showed you in the beginning that what we know from human disease is, of course, a very chronic state of disease. So all those features I introduced in the beginning are from a chronic stage of disease. And this is here in the graph shown in light gray. So you have this sort of exponential growth of the human aneurysm. And then looking at the 4 different mouse models that I just introduced, sorry to go back one slide, you can see that they all at different time points. First of all, none of these actually grow in an exponential way. They would all grow in this more linear way and actually lose speed again. And from very different stages of those models. So time point 0 is the induction of the aneurysm, and then you have the course of 4 weeks. You know that there are distinct changes in the PPE model going on happening here. There are distinct changes in the angiotensin model in a timely manner. There are very distinct changes also in the calcium chloride model and also in the remaining models. I'm not going into detail here. You can all read this in the manuscript, and of course, I'm also happy to answer questions on these, but something that is very important, what we think is to actually choose the right point for intervention in using those models to actually apply some medications here. So I guess most of you are aware that most studies actually induce the aneurysm and then very early also start to give some sort of drug to start some sort of treatment, which basically in most of the models just means that you basically treat acute inflammation that is happening here, and you don't actually treat the aneurysm features going on here. And we think that starting at later time points, starting at day 7, starting at day 10, you're losing a lot of this aneurysm growing dynamic, but you're treating more accurately actually the features that also happen in the human disease. And I will just take the last slide to show some work. Just before this webinar, actually, we got the confirmation that this work will now also be published soon. So we've been using this PPE model, and you can see the normal growth curve of those PPE mice here. So 4 weeks, about 60 to 70% of aneurysm growth going on here. And we've been treating those mice with lenvatinib, which is a drug already used in cancer treatment in the clinical setting. We're trying to repurpose that to aneurysm based on angiogenesis, based on smooth muscle cell activity. And when we actually do start an oral treatment at day 7, so with this already established aneurysmatic lesion, we can see a nice complete halt of aneurysm growth, basically. We've then taken this also further and started to mimic some endovascular treatment on those mice. So performing a reoperation on day 7, introducing lenvatinib into the already established aneurysm, you can basically see the very same effect, so complete halt of this aneurysm growth. And now coming back to my initial point, so what we see here, we have an aneurysmatic lesion, we treat it, and we see some very nice regression of this aneurysmatic phenotype, and this is of course all based on at different levels here. So to conclude our opinions on the mouse models, as stated in our review, there are a lot of mouse models available. They are very valuable for translational research, but you have to consider that each of them is mimicking a very specific feature of what is probably the nature of human disease. Angiotensin is, of course, best investigated, it's most used. You have to consider that it's a very prominent aortic dissection going on here. The hemorrhage is appearing in the aneurysm wall, actually, or in the aortic wall, and something that's a point that also even Alan Dougherty's group is trying to stress now, that reporting standards should be kept. So we know that in the angiotensin model, there is actually 4 different types of aneurysms, 4 different ways that the aneurysm develops, which is currently not reflected in none of the studies we're seeing or we're reading, basically. So I think this is something that really needs to be considered a bit more. PPE, we think, is a very good mimicry of human disease. Of course, it's surgically challenging. You don't see rupture, you don't see any intraluminal thrombus here. So that's a limitation. Some of that stuff is better covered by putting elastase on the outside of the aorta, actually, where you do see rupture, where you do see some thrombus. So we're curious to see what happens here at different time points and how well this actually reflects what happens in humans. I'm stressing this point a lot. So the specific timeline of the development of the aneurysmatic lesion in those mouse models, that's very crucial to actually look at really translational results here. And I've spared this point. We've done some good work with Goergen from Purdue University to actually do different sorts of high resolution imaging on those mouse models, on those aortas. And we think that those mice can be very nicely used here, and with some limitations also for endovascular treatment. With this, I would like to thank you for your attention, and I'm of course also very happy to take any questions.
Thank you very much for your presentation, Dr. Busch. Very interesting to see. I like this slide where you put all the models together. It was very interesting. I have one question. Do you think that the current available mouse models are sufficient to mimic the human disease? And well, go ahead with that.
Well, yeah, so I've never invented a mouse model, so I think it's quite hard to come up with something that actually would mimic all that stuff. I think what we know from human disease, of course, we don't know anything about the origin of aneurysm development. I think the available mouse models, they all have some very good features, but you would also want to combine 2 or even more mouse models, and I'm stressing this point. So really look at the time point of interest. Of course, you will lose this very dynamic, steep increase in the beginning, but I guess that's something that needs to be considered.
Yeah, it's a good answer. There is no one model that can replicate, or answer all the research questions. So, do you see any place for, or where do you see the place for large animal models in AAA?
Yeah, so we've been using the PPE model in a preclinical pig model as well. So I think this is something that actually has a place also in translational and in basic research, not only for putting different sorts of prosthesis into pigs. So you would also want to have an organism that mimics human a bit more closely. So having the same blood volume, having a bit more similar genetics, and also being able to perform probably some endovascular interventions. So I showed you what we did in mice. So we would also introduce the aneurysm in a pig and then actually put a drug-coated balloon in there and try to influence aneurysm growth in those large animal models, which of course can't be done in mice, or can only be mimicked in mice very limitedly. So I think there is a place, but of course it's cost expensive. It needs well planning. There's ethical considerations, but I guess there will be a bigger and bigger place in the future for that.
I really appreciate it. Thank you so very much for your presentation. Unfortunately, we have more questions, but we will try to direct this to you, but we need to move along right now.